Hi class, it's Jamie Batch, your instructor. We're wrapping up chapter 19 and we just finished talking about labor, including premature labor. Let's skip to screen sharing mode. We can get back to our PowerPoint and into all of the nitty-gritty for the end of the book. So there's the neonatal period, which is after birth into the first 28 days of life. So this is considered the neonatal period. Infancy is the first year of life, and it's dependent on the mother for nourishment, transportation, protection. That should be pretty obvious, right? Milk production. By the end of the 6th month of pregnancy, mammary glands are starting to finalize their development. They may even start secreting colostrum. Colostrum is a special secretion that contains antibodies that will help fight infection until the newborn's immune system is fully developed. A few days into nursing, the mammary glands produce breast milk. So when you first have the baby, there's just colostrum that's coming out. There's no milk yet. Your breast won't feel very full for a few days. And then after a few days, I think with my son, it took maybe 4 days. And then with my daughter, it was just a day. So the more practiced you are at breastfeeding, typically the quicker it will come in. Your body will remember. A few days into nursing, mammary glands produce all that breast milk. It's much higher in fat content than colostrum, and it contains antibodies as well as lysozyme, which has antibiotic properties as well. So breast milk is provided through a milk letdown reflex. The milk letdown reflex is stimulated by tactile receptors, which is the suckling of the infant. This sends a neural impulse to the spinal cord and then to the brain, stimulating the hypothalamic nuclei to release oxytocin. Oxytocin is then released from the posterior pituitary, and then the milk is ejected. Oxytocin also causes contractions of the lactiferous ducts and sinuses to get that milk out. So here's the milk letdown reflex. Stimulation, and then oxytocin is released, and milk is ejected. There are life stages involved in development. There are 5 life stages. We mentioned the neonatal period. This is the first 28 days of life. Infancy is the first year, dependent on the nutrition contained in milk. And then childhood is when the infant is weaned from the breast milk in early childhood, and the body proportions will gradually change. Adolescence begins at puberty, and this is the period of sexual maturation, and it ends when growth is complete, and then finally maturity. So postnatal hormones have several influences, like growth hormone, adrenal hormones, and thyroid hormones. Adolescent growth and development is most affected by the sex hormones, and maturity is when gradual changes with age will take place. And this is that aging process that we pretty much talked about at the end of every chapter. We mentioned the effects of aging. You can pause the video here, go back and review this section. So let's talk about some hormonal changes at puberty. First, the hypothalamus increases production of GnRH. The anterior pituitary is going to respond by increasing levels of FSH and LH in the body. In response, the testes and ovaries are going to initiate gamete production, secretion of their own sex hormones, testosterone, estrogen, and progesterone, which is going to stimulate secondary sex characteristics as well as behaviors. Sudden acceleration in growth rate, ending with the closure of epiphyseal cartilages, will take place as well. So, responses to testosterone in men. Develop of hair on face and chest, terminal hair growth in the axillary and genital area, that's that thick kind of pubic hair and underarm hair, accelerated bone deposition and skeletal growth, skeletal muscle growth and muscle mass increase, activating the central nervous system in the centers that are involved in behavior and sexual drive, increasing blood volume and hematocrit, thickening of the larynx, lengthening of the vocal cords, functional development of all the reproductive organs, and spermatogenesis. This is just a summary of everything that I just went over. Effects of testosterone in men. Responses to estrogen in females. Continued development of fine vellus hairs throughout the body, terminal hair in axillary and genital areas, initial development of mammary glands, more rapid epiphyseal closures, stimulation of some skeletal fibers, central nervous system involving sexual drive and behavior as well in females, decreased plasma cholesterol levels, increased risk of iron deficiency anemia because of iron loss with menses, thickening of myometrium, increased blood flow to the endometrium, and the functional development of all of our accessory reproductive structures. You can pause the video here, go back and review this section. The next part of this chapter revolves around genetics, which obviously there's entire courses dedicated to genetics. This is just the tip of the iceberg. And at this point in the semester, I don't want your understanding of genetics to make or break you. So we're going to stick to some of the basics and not complicate things, as they can be very complicated. So inheritance is the transfer of genetically determined characteristics from parent to offspring through generations, and genetics is the study of those mechanisms that are responsible for those patterns of inheritance. So we have some vocabulary associated with this. Genotype is the chromosomes and the component genes. It's analogous to kind of like the blueprint or the architecture plan of the house. This is what your DNA says. Your phenotype is the physical characteristics or anatomical characteristics that's displayed because of your genotype. This is like what the house actually looks like. So the genotype is like the plans, the phenotype is the actual structure that you see. A karyotype is the entire set of chromosomes. There's 23 pairs of chromosomes. One pair came from the sperm and one pair came from the egg. The two together are called homologous chromosomes. 22 of the 23 pairs are autosomal, these affecting all of our somatic characteristics like hair color, eye color, lactose intolerance, and all of those things. The last pair are the sex chromosomes, which are the XX if you're a female or XY if you're a male. There is a karyotype of a male. You can see right here the XY. One of these chromosomes came from your mom. One of these chromosomes came from your dad. One from the mom, one from the dad, one from the mom, one from the dad. And even though one might look a little more bent than the other, it has the same exact genes in it, maybe just different variations of those genes. So these homologous pairs are lined up, and they each have genes at the same exact location or locus on each chromosome. The two chromosomes may carry the same information or allele, or they may carry different forms of that gene or different forms of the allele. If you have the same allele on each chromosome, we call you homozygous for that particular trait. If you have different alleles for that gene, we'll call you heterozygous for that trait. So here's one trait of this person's homozygous for it. Here's another trait, same chromosome, same locus, but this is a different allele. This is heterozygous for that particular trait. So overall inheritance is when we're looking at the phenotype of a heterozygous genotype, depends on how the corresponding alleles will interact. The most common form of interaction is called simple inheritance. Strict dominance is when a dominant allele is expressed in the phenotype. For example, even if you have only one allele for freckles, you will have freckles. So freckles is the dominant allele over the non-freckle allele. The recessive allele would only be expressed if both recessive genes are present, if both recessive alleles are present, or if you're homozygous for the recessive allele. So there's a cute kid with freckles. We use Punnett squares to help us determine the probability. The dominant alleles are represented by capital letters. The recessive alleles are indicated by lowercase letters. So big T is homozygous dominant, big T is heterozygous, and little t is homozygous recessive. Punnett squares are how we predict the genetic probabilities. For example, if you want to say if a father's homozygous for normal pigmentation, but the mother's homozygous for albinism, all of the children will be heterozygous, but they will show the phenotype of having pigment. So here's how it works. You put the alleles on each column or each row. Each allele gets its own row or column. So the maternal alleles are little a, and the paternal alleles are big A, big A. And then you drop from the top and slide from the side, and you get your probabilities of offspring. So in other words, all of the offspring from this scenario are all going to be heterozygous. In this case, if we have maternal alleles, little a, she is homozygous recessive, but the father is heterozygous, again, we drop from the top and slide from the side. So these two children here are heterozygous for the trait. They have a big A and a little a allele, whereas these children will be homozygous recessive and will have the albinism trait. Polygenic inheritance is when many phenotypic characteristics will interact between several genes. So the resulting phenotype depends on the nature of the alleles and how those alleles interact. This would be like the shading of brown or black hair, our skin color, our eye color. Almost everything in our body is polygenic. Very few traits are only affected by one particular gene. So again, this is why we're all a little bit different, even from your brothers and sisters. You can pause the video here, go back and review. Let's talk about some abnormal chromosomes or genes. Chromosome abnormalities may involve thousands of genes, and they're usually lethal, meaning will end up in, killing you basically. Variations in the structure of individual genes are relatively common. More than 99% of human nucleotide bases are all the same. So you and me are 99% identical. About 1.4 million single base differences or single nucleotide polymorphisms exist. These are typically associated with specific diseases. A trisomy is when you have 3 copies of one chromosome. The most commonly talked about trisomy, and it actually is the most common trisomy, is Down syndrome or trisomy 21, 3 copies of the chromosome number 21. It's the most common viable chromosome abnormality, meaning if you have 3 copies of chromosome 1, that is not a viable life. Will not be born, will result in a miscarriage. Trisomy 21 will result in mental retardation, physical malformalities. Degree of mental retardation varies from moderate to severe. Anatomical problems with cardiovascular system can be fatal. In adulthood, many develop Alzheimer's very early on, as early as age 40. There's a direct correlation between maternal age and the risk of having a child with trisomy 21. The risk increases from 1 in 2,000 to 1 in 900 to 1 in 46. So a very, very high rate as you get older, as the women get older. That's a karyotype of trisomy 21. There's also Klinefelter Syndrome. This is an individual who has an extra X chromosome. So they have a sex chromosome pattern XXY. They will be male because they have that Y chromosome, but will have reduced testosterone production. Testes typically will not form fully. They will be sterile. Breasts will be slightly enlarged. 1 in 750 males will have Klinefelter syndrome. Some males won't realize that they have Klinefelter syndrome until they go to get tested for, infertility, and then they do a karyotype and say, oh, well, you have Klinefelter syndrome. That would be why. So, Turner syndrome is when the individual only has 1 X chromosome. So typical chromosome deletion is called a monosomy. So monosomy or trisomy is 3, monosomy is 1. The phenotype, it would be a normal female. Estimated occurrence is 1 in every 2,500 female births. The maturation changes do not occur at puberty. So the ovaries are non-functionable, very little estrogen. Physical abnormalities include very short stature, low set ears, and a webbed neck. Turner syndrome. Only 1 X chromosome. X-linked characteristics are on the X chromosome. The X chromosome is larger than the Y chromosome. I'll flip back here so that you can see that. Here's the 2 X chromosomes, very large chromosomes compared to this little Y chromosome. So the X chromosome carries genes that affects things like somatic structures. These genes are called X-linked or sex-linked. There's no corresponding allele on the Y chromosome. In fact, you cannot live without an X chromosome. If someone is born with just a Y chromosome and not an X chromosome, well, they won't be born. It would miscarry. That is an impossible, not viable life karyotype. Many associated diseases are associated with the X chromosome. Color blindness is one of them. So again, here you see X-linked alleles not present on the Y chromosome. You can live, I'm living a perfectly normal life without a Y chromosome, but nobody can live without an X chromosome. Color blindness is carried on the X chromosome. Males can have a dominant allele. That would be represented by the letter X for the X chromosome and a capital C, that's showing the dominant allele. Males with the recessive allele, X lowercase c, have red-green color blindness. Females can have 1 dominant allele and 1 recessive allele, so they can be heterozygous X big C, X little c. But females must have 2 recessive alleles in order to have red-green color blindness, X little c, X little c. So since females need to have 2 copies of that recessive allele and males only need 1 copy, things like color blindness and other X-linked traits like hemophilia and muscular dystrophy are more common in males. So here you see this inheritance pattern. Females, X big C, X little c, male, X little c, Y, and so you get this combination of normal females and a colorblind male. There are several tests that can be performed during pregnancy to basically test any chromosomal abnormalities with the fetus, and you can do this if you are predisposed for a condition or if you've been advised maybe some abnormal information came up on an ultrasound or something like that. Amniocentesis, amniotic fluid analysis, or an AFP are all ways of gaining information about the unborn fetus. You can pause the video here. Oh, this is the end, actually. You can stop the video here and answer these 3 questions. Please let me know what questions you have about Chapter 19, the final chapter in our book and the end of our semester. Please let me know what questions you have before you take the final exam as well. I'd be more than happy to review with you. And have a great day.